He's a sports scientist. He's got his PhD. He does incredible research in this field. This dude knows what he's talking about. On this episode of The School Muscle, Dr. Cody Haun. The first thing that I'd kind of like to discuss here is how certain people can do the same exact training but see different results. So kind of this heterogeneity in response in terms of training programs, and there's kind of different terms thrown around like non-responders and high responders, and you've got a few papers out kind of on this. So if you could kind of give some general overarching things on kind of what you found and your thoughts on this, that'd be great.
Really interesting area. Why can multiple people do the same training intervention but see different results? Fundamentally is the question. And we found some pretty interesting stuff, I would say not expected. So you mentioned we've had a few studies now. So to give a little bit of background, I'm at LaGrange College now. I'm a professor, and we are starting up a research arm of our program and hopefully going to be digging more into this. But this work was done at Auburn University with my mentor, Dr. Michael Roberts. And I know he's probably in the lab right now doing some assays that are related to this area. Great mentor, great lab. So we did, like you said, a few different studies, really similar training. So mostly, I would say, hypertrophy-based training. We published a review in the area. And the overarching theme so far, to, I think, make this practically useful, is that we really haven't found many what we would refer to as biomarkers that we can practically track, at least in the practical setting, that are predictive of the hypertrophic response. So, for example, like we've looked at a variety of hormones, we've looked at a variety of messenger RNA molecules from biopsy samples, so actually extracting muscle tissue from the quadricep and studying things at baseline and then after the intervention, both messenger RNA and protein expression patterns. How do those predict how someone responds to the training intervention? We've looked at a variety of those and haven't really found much that's super predictive. Now, one interesting thing that we've found more recently from my dissertation is that, and this work is in review, so this isn't officially published yet, and I hope that it will be soon. It's been in review, unfortunately, for like 5 months.
Yeah.
So we've been patiently waiting, but the preliminary findings suggest that individuals that start with larger muscles from the vastus lateralis ultrasound image, so the thickness measurement, so the thicker the muscle was at baseline, the less the muscle grew. That was in Dr. Brooks Mobley's dissertation where we found that. And then in my dissertation, I found that the larger the fibers were at baseline, so using immunohistochemical staining and looking at muscle fibers under the microscope, and then using software to size like 100 fibers per cross section. So we had 4 time points total. There was 3 in the analysis, so that's pre, mid, and post 6 weeks. 100 fibers. You can imagine how much time this took, and that's part of the reason why it's not published yet, because we finished the study almost a year ago. But 100 fibers per subject per time point. We had 30 subjects, and so that's like
That's ridiculous.
it's like 10,000 or more fibers. So I'm really confident in the analysis. To make a long story short, like we use specialized software to actually size those fibers, and we'll get into some of that, I know, later in the call when we start talking about hyperplasia and potentially fiber splitting and stuff like that. But found that the larger fibers at baseline grew the least, and therefore like the smaller fibers grew the most. And it's not the most groundbreaking finding, but I think it's relevant nonetheless. And that is to say that individuals who were bigger at baseline grew less in response to the intervention, at least at the fiber level. And we also found in that analysis that individuals who started with more type 2 fibers, so of those cross sections that we analyzed at baseline, individuals that had a greater proportion of type 2 fibers, so not getting into type 2A and 2X, and a reviewer was correct in pointing out that we really need to understand if these fibers were 2A or 2X to better predict the response. Because I know you had Andy Galpin on recently, so shout out to Andy for his work with fiber typing. I know he would punch me in the face if I just said there were one type of 2 fibers, and he's all into the pure and the hybrids, and I think it's awesome. And that's really the most appropriate way to analyze fibers and type them, is at this single fiber level. We found that individuals with greater numbers of type 2 fibers at baseline grew more in response to the intervention than individuals with lower amounts of type 2 fibers. So to summarize, because I know that was a little bit scattered, the larger the fiber was at baseline and the larger proportion of the fibers in the cross section of our microscopic image, the more predictive the response. So the larger the fibers, the less they grew, and then the more type 2 fibers, the more they grew overall. So those are, again, preliminary findings. Hopefully those are published soon. There's a ton of other biomarkers in the paper, but that was what we found in this analysis. And like I said, other than that, not much predictive of the response, at least the training interventions that we used. Now, I've said this before on other podcasts, but I don't think that it's fair to characterize an entire human as a responder or a non-responder off of a microscopic image or one measurement. And it's not correct to say that because someone didn't respond to one intervention, they're not going to respond to another one. So they may have been a non-responder to the intervention that we use, but that doesn't mean that they're not going to respond well to another one. I know other people have argued the same thing, but I do think that that's an accurate representation of the evidence as a whole, that a response will happen to different degrees and in response to different interventions for different people. I know that's sort of hand waving, but nevertheless, did all that make sense?
Yeah, that made sense. And, part of that is actually kind of counterintuitive to me, because something you mentioned was that the more proportion of type 2 fibers grew more, but the individuals that started with larger fibers grew less. But my expectation, I guess, would be that the individuals starting with the larger fibers would also have larger proportions of type 2 fibers. Does that kind of get what I'm saying there?
Yeah, that's a really excellent question. So again, I can't go into like a ton of detail on the analysis. When it's published, we can have a whole another podcast just talking about that paper. But to make a long story short, we basically used type 1 staining, and anything that was negative for type 1 staining, we called type 2 fibers. So therefore, type 2A and 2X were lumped together.
Okay. And
then the type 1 fibers were green in the cross sections. And so a way that we commonly express fiber cross-sectional area at any level of time when we do immunohistochemical staining is what's called the mean fiber cross-sectional area. And it's not just the average of all of the fibers in the cross-section, which is a fair way to do it. So like if I had 150 fibers in the cross-section, then take the average of all of the fibers in the cross-section, right? That makes sense. Mm-hmm. But when you're looking at a 10x magnification, for example, it's important to weight the number of fibers by their type. So like of the 150 fibers, if I have 100 type 2 fibers and 50 type 1 fibers, is it fair just to take the average in general, or weight the measurement based on the fact that I had double the type 2 fibers. So that's based on mean fiber cross-sectional area, just to kind of make a long story short. And again, like the R values weren't super strong. I mean, we're not talking about like this groundbreaking finding that everyone should get fiber typed at baseline, and that's going to predict how people respond to training. If memory serves, the R values were like 0.4 or like 2, I think was one of the highest. So, I mean, it's not that large of an explanation, but it was larger than the other biomarkers that we analyzed. And we analyzed a ton of them. So like a ton of hormones, like I said, a ton of mRNA, a ton of proteins from the biopsy samples. And yeah, so I think it's also important to mention in that paper, we used a composite score of hypertrophy, which we can talk about this more when we get to the hyperplasia related questions that you were wanting to discuss. Mm-hmm. But we felt that, and we did this in another analysis that we performed, where it's, again, probably not appropriate to classify someone as a responder or a non-responder based on one measurement. So what we did was actually create a composite score of multiple measurements, where we had the fiber cross-sectional area measurement, which I think will help from what you were saying. Like, it's kind of confusing how that actually worked and why the larger fibers versus the type 2s, that doesn't really make sense. So if we take a fiber cross-sectional area value, a muscle thickness value, a DEXA upper leg lean mass or soft tissue mass value, and a thigh circumference value, and we average them or sum them to form a composite score, sort of like when you take you probably took the ACT or the SAT, right? And you get this composite score. So like a composite score is hypothetically representative of your overall knowledge, right? Right. And so it's similar to this variable, which we called hypertrophy, but it was really a composite score of multiple measurements. So when you zoom all the way into the single fiber level, we're looking at fiber cross-sectional area. If we go up a level, now we're looking at vastus lateralis thickness. If we go up another level, we're looking at DEXA lean mass. If we go up another level, we're looking at thigh circumference. Does that make sense?
Mm-hmm. Yep.
And so we used those values and how they changed over time to create a composite score. And not saying that that's a perfect method, we just felt like that was a reasonable method in hopes that better represents the change in muscle size, and you could say hypertrophy, which we'll unpack later. We felt like that was better representative of the adaptive response than just one measurement like we had done previously.
Something that is kind of along the lines of these measurements is that sometimes when you measure something on a more microscopic level, it's not necessarily what you would expect on a more macro level. So could you kind of go into that a little bit, how sometimes what you see on kind of the macro isn't what you're seeing on the micro and what might be going on there?
Yeah, so I just spent with, like we talked about a little bit before the call there, about a year working on a review article that I hope will be published in the next week or two. And since we'll mostly be talking conceptual, I'm happy to share most of the key findings. We started to find that, well, in our own studies, fiber cross-sectional area changes didn't always agree with ultrasound changes or DEXA changes. And we were puzzled, frankly, because the working model of hypertrophy is that the changes in muscle fiber size and we're talking about in context of resistance training, right? So the changes in muscle fiber size are primarily due to increases in myofibril size. So those cylindrical thread-like structures that run through a fiber, those will either increase in size or number. So an increase in myofibrillar size would be probably caused by an increase in the number of sarcomeres in parallel, right? That's sort of the hypothetical model. and that would increase force production. And then it's commonly argued that an increase in serial sarcomere number will increase velocity of shortening, right? I'm sure you've heard that in grad school. And that's what I had always been taught. So that can happen. And then increase in sarcomere number in those myofibrils can ultimately increase the size of the fiber, right? Because a muscle fiber is about 70% or so myofibrils. And then you have obviously the mitochondria and then the sarcoplasmic reticulum that make up most of the rest. And then, of course, the sarcoplasm. So the model is as the fiber grows through myofibrillar number or sarcomere number in existing myofibrils, so those are called myofibrillogenesis or sarcomerogenesis. As that happens, the fiber grows. As the fiber grows, the whole muscle grows, right? Or the fibers that compose the muscle. And so as the whole muscle grows, of course, the mass of that muscle and the leg should also increase, right? Mm-hmm. In other words, we would expect these measurements to be correlated. Right. So as the fibers grow, muscle thickness should grow. As muscle thickness expands, muscle mass should increase. As muscle mass increases, then we should see an increase in the circumference of the muscle, hypothetically. Right. But we just weren't finding that. And in fact, we were finding pretty weak correlations. And so this is about a year ago, and we're just like, did we do something wrong? Wondering what's going on. And so I started talking to some other researchers. So a few shout outs, Brandon Roberts at UAB, he's a postdoc, good friend of mine. Andrew Vygotsky, he's at Northwestern, I think, right now, finishing his PhD. Brad Schoenfeld, I'm sure you know Brad. Mm-hmm. Brad's awesome. He was really helpful in the review. And then, of course, my PhD mentor, Michael Roberts, and then my really good friend, Chris Van, who's finishing his PhD at Auburn. Brandon, actually, and Andrew, well, really all those guys would be good to get on the podcast, man.
I just wrote down all of their names here. So.
Okay, good, yeah. We all worked together. We started talking, and we all worked together on a review article. And we've been working on this thing for, like I said, about a year. And it's a beast, but to summarize some of the key things that we found related to my original points, the disagreement in these measurements, we started to go back to some of the historical literature and sort of started with the law first mentioned. So what's the first ever study that looked at muscle growth at the fiber level? So actually extracting tissue from an animal or a human in response to some kind of training intervention. And so found a paper in 1897 from, and you could tell me if you want me to skip to the key points. I don't want to bore you with some of the stuff, but I think it's absolutely fascinating. Yeah.
I think so too.
The name of your podcast is School of Muscle, so this should be like right up your alley.
For sure.
So this paper found that in dogs, when one muscle was removed from one of the legs and the animals were subjected to a period of weeks of run training, the other muscle of the other leg grew significantly because it was having to take up for, the loss of muscle mass in the other leg, right? Okay. And that's sort of like the synergist ablation model in rodents that's commonly used to study hypertrophy mechanisms. So, of course, the mass of the other muscle in the other leg grew. And Morpergo actually did some staining and found that, at least in his interpretation of the data, that sarcoplasmic expansion, not myofibrillar density, better explained the increase in fiber cross-sectional area of the muscle that grew. So you have heard, and I'm sure a lot of listeners have heard of sarcoplasmic hypertrophy versus myofibrillar hypertrophy, right? Mm-hmm. That's kind of been speculated for a period of years. And with no clear, I would say, at least this was before I did the review article, it was unclear if either of those was a thing, but probably not. It was probably primarily a myofibrillar, process, or at least a myofibrillar-driven process. Well, anyways, so what was intriguing to me when I found that paper is like, wow, the first paper, even though it was in animals, found that maybe the increase in muscle fiber size was due to an increase in sarcoplasmic volume. And so this is around the same time that the Peer J paper was being worked out, which we basically studied sarcoplasmic and myofibrillar protein concentration changes in the biopsy samples after my friend Brooks' training study. So to summarize the basic study, untrained guys, 12 weeks of training, biopsies before and after, and we clustered some of them into a group of low responders where we didn't observe much of an increase in cross-sectional area, and then we clustered another group that we saw this significant increase in cross-sectional area. So we took those two groups of biopsy samples, and we wanted to see how did the sarcoplasmic protein concentration change in each of the groups, and then how did the myofibrillar protein concentration change in each of the groups. And so sort of to back up and zoom out for a second before I start talking about some of the other authors' findings along the years here, the model would suggest that the stoichiometry of the muscle fiber would be maintained. And that is the myofibrillar protein concentration as the fiber grew should be maintained.
So it would like grow proportionally then?
Exactly. Right.
Okay.
Yes. Really nice insight. So the other model would suggest that there would be a significant increase in myofibrillar protein concentration, right? So it's either going to be maintained or there's going to be an increase in myofibrillar protein concentration.
Makes sense.
Yeah, the working model, again, which suggests increase in myofibrillar number or size, causing an increase in muscle fiber cross-sectional area. So it doesn't suggest an increase in sarcoplasmic protein concentration or volume. So we found that there was no significant difference in sarcoplasmic protein concentrations between the clusters or myofibrillar protein concentrations between the clusters. So we found that neither one of them significantly changed. So, I mean, we're just like, okay, did we do the assay wrong? I mean, like I said, Dr. Roberts was the one that really, and he's basically, we called him like, the wizard and the pipette whisperer and all sorts of things because he's just a master at assays. And he spent weeks like optimizing this thing, and Matt Romero and really the team trying to figure out, okay, did we do these right? And to make a long story short, the validation studies that we did to ensure that the data were sound suggested that they were, that they were sensitive enough to pick up pretty small changes in protein concentration. And so, we're like, well, I mean, what is it? And so the next culprit is mitochondrial volume or sarcoplasmic reticulum or T-tubular volume, because those are the other two primary constituents of the muscle fiber.
Right.
And not only in this Peer J paper, but also in my dissertation, and these data will be published soon, it seems that mitochondrial volume decreased. That's based on citrate synthase activity, which is a pretty good method of, estimating mitochondrial volume. As citrate synthase activity increases, there's good transmission electron microscopy data that suggests that mitochondrial volume will increase. So if it's not mitochondrial volume, then the remaining idea is that it's the T-tubules, the sarcoplasmic reticulum, and we're talking about a short-term intervention. I want to be really clear about that, and we can talk more about this in a second. Like, okay, so what did the review find? Blah blah blah. But there are other possibilities. It's certainly possible that the training intervention that we used caused these protein level and morphological changes. So I'm not saying that what we're finding is like a game changer and myofibrillar protein doesn't increase, sarcoplasmic protein concentration doesn't increase. That's definitely not what I'm saying. But from this study, 12 subjects per group, roughly, so it's limited in scope, but we didn't find that changes in myofibrillar protein concentration or sarcoplasmic protein concentration or mitochondrial volume seemed to explain the change in fiber size. Now, what's the other thing that could change? Well, we know that, like I said, sarcoplasmic and T-tubule volume, but fluid, right? So, and ions. So, like more, and that's osmosis, right? If more ions are in the cell, then more fluids in the cell. Or during states of edema, perhaps that because the extracellular fluid is so high, there's a compensatory effect of increasing intracellular water volumes. And that's what I found in my dissertation, that as training volume increased, so did the extracellular and intracellular volume of fluid. So maybe it's fluid, right? And an ion concentration. So these are things that we're following up on. And I think you read the study in Frontiers, like the first paper for my dissertation, showing that as the volume increased past like 20 sets a week per muscle or per movement up to like 32 sets a week, that the DEXA lean body mass corrected for extracellular water change, hypothetically due to swelling-induced edema. It was still significant from 10 sets to 20 sets. So it's still over a kilo when you remove the change in extracellular water from that lean mass value. But when you move from 20 sets to 32 sets, there starts to be some differentiation there, right? Where more of the change in lean mass observed was explained by extracellular water retention. So it's important, I think, to correct for fluid. And in our study, that's a limitation, right? We really, and it's not easy to measure, right? Like basically you have to, I should probably pause here. Am I good?
Yeah, I think we're on track. We're good. I'm loving every second of this for sure.
Okay, cool, man. So we didn't correct for fluid in the Pure J paper, and we're hopefully going to be publishing this data within the next couple of months. This was a really, really involved series of experiments where you basically take muscle biopsy samples, you weigh them in a hydrated state, and then you dehydrate them, and you calculate the difference. And that tells you the essentially water composition of that biopsy sample, right? Mm-hmm. Now, unfortunately, that doesn't tell us if that water was inside of the cell membrane or outside of the cell membrane. So we're still left with that limitation. And we have some ideas of ways to improve our understanding of that. We worked with Dr. Jordan Moon from ImpediMed. He's an expert in bioimpedance spectroscopy, which is like a more advanced method of looking at BIA. So it's similar. It's not the same, though. There's a lot more frequencies involved with BIS. And so they've started to work on sort of a local analysis of intra and extracellular water, so that hypothetically we could use this device to look at from around where we took the biopsy sample, what were the intracellular and extracellular water amounts at that sampling time point. So that's something that we're working on. But to go back to the wet weight and dry weight biopsy stuff, so did that make sense? Like we take the wet weight, we dehydrate it, and then we now have a dehydrated sample of muscle tissue.
Yep. And the difference is how much fluid was in the...
That's right. Yeah. So we're looking at all that, and we've really fine-tuned. And again, I want to shout out to Dr. Roberts, because like I said, I probably could call him right now, and he's in the lab pipetting. I'm not joking. He's been working really hard on fine-tuning the methods to extract myofibrillar protein. And the most recent data that he sent, like he's doing a great job of it. Like we're getting pretty pure samples. So I'm really confident that our next study in the area will help provide some clarity, at least what happened in response to the intervention that I used for my dissertation. Now, to segue into the review article, I know that was a lot to say, perhaps a little here, but we're not the first group to find this. And I'm talking about this, not only you with you on the podcast, but I've been talking about this now for months with other researchers, and people are experiencing similar problems in other labs. And we're thinking similar things that we were, like, maybe it was us. Maybe we did the assays wrong. Maybe something's off and weren't comfortable, following up or reporting that data. And what we found when we went and we reviewed the literature, so just to give you an idea, I mentioned the Morpurgo study in the late 1800s. If we jump ahead to 1967, there's a paper from Gordon that essentially said, I think the title of it was like, "What is hypertrophy?" And he talks about how he was finding that hypertrophy wasn't necessarily the sum of its parts, that the change in whole muscle size wasn't predicted by changes in muscle fiber size in his own experiments. This is in the 60s.
So he was already seeing some of this, what the hell's going on stuff.
Yeah. And so when you jump ahead to 1969, 1970, and so around this time, Bergstrom developed the biopsy technique. And then eventually we developed like a suction modified biopsy technique, which is what we used at Auburn, where you have the needle in the leg after a series of steps to make sure everything's sanitary. It's an outpatient procedure. It's not a big deal. But, we have a suction device connected to the needle, and we extract muscle tissue, right?
Mm-hmm.
So that technique was developed in the 60s. And then Penman in 69 and 70 was the first to study these microscopic changes to muscle tissue in response to training interventions. And so without going into the methods of his paper, he used what's called transmission electron microscopy, which is like incredibly painstaking, where you are like literally zoomed into one single fiber if you want to be. And he counted the density of myofibrils in individual fibers.
Dear God.
Yeah. So like this is like pre-internet days, right? So like this is like what he was doing for fun, I guess. But so like come on, that's admirable. And so that even though that data is old, it's still really useful and it's still really interesting. I mean, you go back and read some of this stuff, it's fascinating. And he found in both of those studies some interesting things. In some, because he did some kind of what we would refer to as odd training protocols, and they were pretty intense, like 5 or 6 days a week, pretty intense training. And in the first study, he had like an isotonic condition, isometric, and then like a run group. And then in the next one, he had a combination of lifting and running for like 5 days a week. And he found, in general, that myofibril packing, so that while the cross-sectional area of a fiber actually either stayed the same or decreased, the number of myofibrils in that area of the fiber actually seemed to increase. And so he basically counted the number of myofibrils in like an area of 5 microns squared and found that the number of those increased in response to the intervention, but the size of the fiber didn't increase. So we've now referred to that as myofibril packing because they got stronger. But now we know that, okay, it may not have been like a local change. It was probably more neural, and that's why they got stronger. Anyways, that was his interpretation of the data. But nevertheless, it's odd, right? Like you don't hear that term a lot, myofibril packing.
Right.
And you don't hear data often that's like the fibers didn't grow that much in response to a 12 or a 16-week training intervention. But that's what he found. So you start moving along through, and I'm not going to go through like every study because there were quite a few. You start to find these disparate findings where biochemically and ultrastructurally things weren't necessarily lining up all the time, at least according to the model. And that is an increase in fiber size due to an increase in myofibril number or myofibril size. So through the through the 80s and the 90s, indeed, we found in general resistance training increases muscle fiber cross-sectional area, right? Like that was a theme through that couple of decades. And that was really the primary research being done, is that taking a biopsy, doing immunohistochemical staining, studying how the fibers changed in size, and fiber typing them. And through that time, it seemed to be that type 2 fibers, or primarily type 2 fibers, I should say, grew or were more responsive by comparison to type 1 fibers. Like that was the general theme through that era. But what kind of got lost in that era, or really just wasn't investigated commonly, is myofibrillar protein concentration changes or sarcoplasmic protein concentration changes. Now, there was one study, and this is the important one that really led us to start digging into this even more recently, and that was in 1982 from McDougall. And this is a really cool one. And he found that after 6 months of training in untrained individuals, that the sarcoplasmic fraction, using transmission electron microscopy imaging again, he found that the sarcoplasmic fraction of these muscle cross-sections increased significantly, okay, in response to 6 months of training. So not just short-term interventions, because a lot of those studies I mentioned previously were short-term. He found that the sarcoplasmic fraction increased significantly. And then he compared some of those findings to bodybuilders and powerlifters, some of which reported being on anabolic steroids, so that's somewhat of a confounding variable. But he found that the bodybuilder and powerlifter fibers had significantly higher cytoplasmic or sarcoplasmic fractions than the control or the untrained group that just went through the 6 months of training. In other words, like that's more of a subchronic group, but still you see this sarcoplasmic expansion. And then he took this group that had been training for years, and he found an even larger proportion of their fibers was composed of the sarcoplasmic portion. But we would expect, based on our model, right, that the myofibrillar portion should be higher.
Right.
Right? And they had significantly larger fibers than the untrained group. So all that being said, it's not to say that myofibril number or myofibril protein concentration didn't increase or didn't change. It's just to highlight the fact that the sarcoplasmic portion is responsive to training and can explain changes in muscle fiber size. So saying that sarcoplasmic hypertrophy isn't a thing, this is stopping in the 80s. Now I can keep going because there's more findings, and there's findings from our own lab that suggest that this is a thing. But sarcoplasmic hypertrophy, and that is an increase in the amount of space that the sarcoplasmic amount or the sarcoplasm occupies in a cross section, it can increase. And the myofibrillar fraction can either be maintained or even decreased in response to training. But if the overall size of the fiber increases, and that's all that we look at, we just look at that 2D cross-section on a microscope, then we don't quite fully get the whole picture and understand how those changes can affect function. And I think that's just an important point to sort of pause and let marinate for a second, that we can't just look at fiber cross-sectional area to understand how certain training interventions will affect function or what's going on at the protein level. So I can stop there, and then we can talk about some of the more recent work that's actually looked at myofibrillar sarcoplasmic protein concentration changes if you want, but we can talk about something that you might have a question about.
So I'm not even going to attempt to try to summarize what I just heard, but it sounds like that there still seems to be a little bit of a discrepancy between what's happening on like the myofibril level and sarcoplasmic level compared to what we're seeing like on the whole muscle level. And it sounds like in chronic training and longer term training, it seems like this sarcoplasmic hypertrophy is a little bit more indicative. That wasn't a word, at least I didn't pronounce it correctly, of whole muscle cross-sectional area. Is that on the right track?
Well, so I don't think that that's an incorrect interpretation. So we've kind of stopped in the 80s, right? When you keep following this data along, it gets more weird. So, like, I can highlight some of the key differences and the discrepancies, but I think it's safer to say, in some, and based on those earlier studies and the duration of those interventions, that some of the short-term responses to training that increase muscle size as measured by fiber cross-sectional area are, if not primarily driven by, at least somewhat driven by, increases in volumes of the muscle cell or fractions of the muscle cell, organelles, fluid, etc., that are not myofibrils alone. So that fundamentally disagrees with our model.
Mm-hmm.
Now, that's the short term. Others have referred to this as preparatory hypertrophy. I think that's a very plausible hypothesis, that some of the short-term interventions, especially in untrained individuals, and a lot of that earlier work was in untrained individuals. In untrained individuals, there are likely, and this kind of agrees with other models, right, that suggest hypertrophy lags, right? Strength adaptation in the early phases are neural. You've probably seen that representative sort of paradigm.
Yep.
Perhaps there are metabolic and morphological alterations that occur early on, maybe 3 to 4 months, and perhaps a period of months, and according to that McDougall data, up to 6 months of potentially observed hypertrophy, right, where the size of the fibers increases, muscle mass increases. And we know that that happens in untrained individuals, but it seems that not all of that increase is explained by myofibrillar hypertrophy alone. I think it's entirely plausible to suggest things like the sarcoplasmic reticulum and the T-tubules can increase in volume.
Mm-hmm.
I think it's entirely plausible to suggest that fluid, intracellular fluid, can increase, and the amount of ions, calcium and sodium, et cetera, and potassium, right, and the exchange thereof becoming more dynamic. A lot of those ion fluxes and ion concentrations can change in such a way that we could start to see fluid shifting and things changing in organelle concentrations, things like ribosome biogenesis, we know that's a thing, and other things that are occurring to increase the size of a fiber that are not, driven by myofibrils alone. So I think that was clear, right?
Yep.
Okay. So we could call that like preparatory hypertrophy. That's setting the stage, that's laying the foundation in a way for the subsequent growth, which I also think is entirely plausible to expect and inform people that, yes, it's likely going to happen that you're going to increase the number or size of myofibrils, and therefore eventually strength driven by that myofibrillar hypertrophy, which I know is sort of a separate conversation. But I don't think that it's correct based on the evidence right now to say that myofibrillar hypertrophy isn't a thing and that you don't eventually start accruing myofibrillar protein and myofibrils. I think that eventually happens. I just think that we don't have enough longitudinal data with standardized training interventions that allows us to confidently say when that actually starts dominating the morphological changes in the muscle.
I think that's clear. And if I don't jump into this right now, we're going to run out of time. So something I have to touch on is muscle hyperplasia. And how does that potentially kind of play a role in some of the things we've been kind of discussing today?
Yeah, so this is a fun one. And we're working on a little commentary on this now, because for years now, we've kind of thought that hyperplasia isn't a thing in humans, that hypertrophy explains the changes that we see in muscle size throughout a training career, right? And that hyperplasia only exists in animals. And some people would argue that, I think Jose Antonio, for example, has written some stuff on hyperplasia still being a possibility, if not a phenomenon observed in human fibers, and Gagne and others. But nevertheless, hyperplasia, from a technical definition standpoint, refers to an increase in fiber number. And in my opinion, that dictates that we're talking about an entire new fiber that is capillarized, that's innervated by a motor unit, that is joined to the tendon, and that is essentially functioning as an entire new fiber, right? With its own organelles, with its, again, own intervention or own innervation from a motor nerve, etc., right? That it's a fiber that consists of its own myofibrils, right? Mm-hmm. From, you could say, origin to insertion, and it's wrapped in connective tissue. To me, an increase in the number of those as an adult would show hypertrophy or hyperplasia. So does that make sense?
Yep, that makes sense.
Now, of course, hypertrophy refers to an increase in fiber size. And we didn't really get to go into too much of the technical definitions that I think we should start to apply to hypertrophy, so I'm going to plug those right now. I think that we should start to use types of hypertrophy to better explain the adaptations that we see in muscle size. So things like a connective tissue hypertrophy or an expansion of the extracellular matrix. We should characterize myofibrillar hypertrophy, where we actually quantify either the number of myofibrils or the concentration of myofibrillar protein that's concurrent with an increase in fiber size. And we should start to refer to another subtype of hypertrophy as sarcoplasmic hypertrophy. So just so we're clear there. But each of those hypothetically could cause an increase in
size,
which would be hypertrophy. That's hypertrophy. Now, fiber splitting, and shout out to Kevin Murak and team from Kentucky. They just published a really cool paper on this, and he would be a really good person to interview too. Kevin's really smart, and he's I haven't heard him on many podcasts, and he would be really good to interview, man. I got to meet him in San Diego, and I think we talked for like 2 hours at a poster. But he would be good to interview, man. And John McCarthy, that's his mentor there at Kentucky. So they showed pretty clearly that fiber splitting, so an existing fiber splitting into, you could say, seemingly two separate fibers, right? That's an actual thing that can happen in animal models. And in their hands, when they use this synergist ablation model or extreme loading protocol, they'll see fiber splitting occur. All right. So think about a tree with a trunk and you have branches, right? But you don't call that tree two separate trees. That's a tree with branches, right?
Right.
So to me, fiber splitting is analogous to a trunk of a tree eventually growing into multiple branches, but that's still one tree. And so I don't think that that's properly referred to as hyperplasia. Now, this seems like semantics. It may not seem helpful to anyone other than us, ivory tower academics with our pencil necks and our white coats in the lab, right? But I think it's important to operationalize the definitions. So I think fiber splitting refers to that. Hyperplasia refers to an entire new fiber that's generated. Mm-hmm. And then hypertrophy refers to the growth of existing muscle fibers,
which consists of sarcoplasmic, myofibrillar, and all these other things that could lead to actual size growth.
Yeah, and I think right now it's fair to say, and those are just positive definitions, and we talk about some of that in the review article. Can we improve on those? Yes. Is this the golden ticket? I'm not saying that. But I think that this is a really, really great opportunity for us as young scientists, people like you and I. Now I'm older now. As you can see, I'm balding and I'm already getting wrinkles. So yeah, I think it's a really, really good opportunity for us as young scientists in the field to dig deeper into these problems, man. I mean, had the way paved for us by these giants in the field that have really helped us understand a lot of things. But I think now is the time where we start asking some of these deeper questions that we can answer with more advanced techniques like proteomics and transcriptomics and things like that, and understand things at a deeper level. But really go back and question some of our fundamental principles or things that we would consider as like absolutes in our field. It's not to say that we, totally try and reinvent the wheel, but let's make sure that what we think happens actually happens.
Okay, so with everything we talked about today, and it kind of seems like the model of hypertrophy as the traditional model might not be necessarily what's actually going on. So with that, are there practical implications to where we may adjust our training in the future to incorporate this potentially new model of hypertrophy?
Very good question. So I don't think we know the answer to that yet. I mentioned before the talk or for the conversation here, like my dissertation data and how it seemed that, fiber cross-sectional area went down and then it came back up. And that's what we found, period. Like in the microscopy images, 3 of us literally sat down after writing code in the software and really trying to be as stringent as possible with how we sized these fibers objectively. They went down in size, and we double-checked, triple-checked all of the images because we were like, this is very weird, because they were going down pretty big time, right, in cross-sectional area. And then they seemed to come back up, sort of that SRA curve. Mm-hmm. But in this supercompensation effect of fiber size, but that's never really been clearly shown, at least in human fibers. So it seemed that they went down and then went back up. But to relate to our fiber splitting conversation, it's possible that fibers that we sized at baseline split into smaller fibers, giving the appearance of a decrease in CSA and an increase in fiber number in that 10x frame, which is what we observed. Mm-hmm. And then those now split smaller fibers grew into larger fibers, which gave the overall appearance that fiber CSA kind of stayed the same, but decreased and then increased. So I'm not saying that's what happened. It's just possible. So to answer that, we'd have to do serial cross sections. And I didn't even mention the Erickson study in 2006. And shout out to Chris Van because he's the one that found that. And they seem to think that fiber splitting was occurring in these higher level powerlifters that were, they're chronically lifting heavier loads, and some of them even reported androgen use. So that's a confounder. But they found evidence of fiber splitting with these serial sections. So, that's another possibility, right? That could be messing with our understanding of the things that I was talking about with the observations with myofibrillar and sarcoplasmic protein changes and stuff like that. But, I didn't share really any of the data after the 90s. So to sort of just give a very brief overall picture of that, it's sort of been all over the board. But by comparison, we have found papers that suggest either a maintenance of myofibrillar protein concentration or a decrease in myofibrillar protein concentration after short-term interventions in both untrained and trained individuals, whereas a handful of researchers have found an increase in myofibrillar protein concentration. So does that make sense?
Yep. So quite a few of the studies show kind of that pattern that you're talking about that might be occurring with maybe it was that splitting that happened and then that kind of that super compensation, but just a handful of kind of showed maybe that's not the case.
Yeah, and that there was an increase in myofibrillar protein concentration coincident with an increase in cross-sectional area. Mm-hmm. So yeah, man, I hope that wasn't too much in like an hour. I know we kind of skipped around there a little bit, but I think there's a lot of questions left to answer. I think that hypertrophy is a construct, and that's what we argue in the review. It's a biological construct. If you look at the etymology of the word, it means excess nourishment. That doesn't necessarily tell us much about what's happening at the single fiber level, right? And when we start to dig into analyses at that level, we really have a lot of work left to do, in my opinion.
Yeah, it's definitely going to be interesting for me to see what kind of comes out about this. If recommendations, like practical recommendations of, do you now train your fibers for this splitting and do that sort of thing? How do you approach that? I think there's a lot of questions to be answered here.
Yeah, Ryan. So, and I'll get into this quickly. I mean, we're doing follow-up research on this, right? So like the traditional speculation, like if you look in Science and Practice and other historical textbooks, like Mel Siff talked about this and others, more practitioners really have argued that higher intensities of load lifted chronically will result in myofibrillar hypertrophy, whereas light loads for high volumes will result in sarcoplasmic hypertrophy, right? So think mechanical tension-based methods versus metabolite accumulation-based methods, for example. So they've argued that happens, right? That with heavier loads, you'll see myofibrillar hypertrophy, lighter loads, high volumes, sarcoplasmic. But I think just to zoom way out, like I mentioned Brad Schoenfeld earlier, I mean, he's done a ton of research on how training volume affects muscle growth and how other variables affect muscle growth. So some things that we know for sure that I think are really safe right now to hang our hat on. One, training with higher volumes over time is likely going to result in more muscle growth. That's a safe bet. And he's even stratified that into set totals per muscle per week. I mean, that's about as practical as you can get, right? Where less than 5 sets doesn't result in as much as 5 to 9 or 10 or more. Right. And you see almost a doubling when you move from 5 to around 9 or 10, and then when you go from 9 or 10 to 10 or more. So I think it's safe to say, like, let's start around there. And Mike Israetel talks a lot about this, right, where around 10 sets and the debate that just happened, starting around 10 to 10 or so sets at the beginning of a mesocycle and working up to around 20, but potentially more, will probably produce more hypertrophy. And we need to start to figure out some practical identifiers of where the point is. We need to cut the block short and deload to hopefully, not sustain an injury and stuff like that. But yeah, man, there's a ton of stuff left to answer, but I think that's a really practical take home for listeners. Training a muscle 2 or 3 times a week is a safe bet. We're doing some research too. So Chris Van's going to do his dissertation looking at higher intensities of load versus lower intensities of load, and how that could affect myofibrillar versus sarcoplasmic alterations. Really cool. So that data will hopefully be published within the year, and we're doing some other work in that regard. And then we're also looking at with Brad some fiber type specific hypertrophy responses. So does the magnitude of the load affect the growth of specific fiber types? And that Bornson paper that you mentioned earlier, like before the call, the BFR study, and Greg Knuckles and company reviewed this one in Mass, which is where I found it, and it was a really cool paper, the Bornson paper, where they introduced some BFR training and found that the type 1 fibers seemed to preferentially respond to just a little bit of BFR training. I think it was like 2 weeks worth of BFR training in the course of like 6 weeks. So that's pretty fascinating. Anyways, man, we could talk for another 2 hours. I said, I called my wife before the call and told her I'd be home like 20 minutes ago. So hopefully she doesn't kill me. I really appreciate you coming on. I appreciate your time. Send the listeners where they can find you, find some of your papers and all that sort of thing. Sweet, man. Yeah. So I have a Twitter now. I have an Instagram. I put some science-related stuff on there and some opinion-related stuff on there. So follow me there at Dr. Cody Haun. I am on ResearchGate, of course. So like research stuff, I try to keep ResearchGate totally up to date. I'm doing some writing for Greg Knuckles, and I work with Greg Knuckles at Stronger by Science as a coach. And he puts out great stuff. Mass is an excellent research review. I totally would encourage your listeners to do that. But yeah, I'm listed as a coach on there. And then I'm also of late serving as a consultant to a company called Aplift. And Uplift is a pretty cool company that's working on sort of optimizing how we deliver training programs, like expert written and such programs to people from really any category of sport performance or fitness that want to achieve a certain goal. So linking people with good coaches and then optimizing the monitoring of data and the delivery of a training program. So they're doing some really cool stuff that I'm providing some consultations to them to help optimize that process, because I've been working in Google Sheets and spreadsheets for years and years, man, and I've kind of always wanted a consolidated, really well done portal and mobile application that I can rely on clients to actually enter data for me consistently. And
that'd be great.
Get that data in front of me and analyze it, creatively, and see the information I want and optimize my workflows as a coach. And that's their vision. So I've been working with them for about 6 months and trying to optimize how all that works, and I'm really enjoying that. But yeah, so in summary, like ResearchGate for research. I'm on Twitter, Instagram. I have a Facebook too. Stronger by Science with Greg Nuckols as a coach, and then Uplift. I'm listed as a coach on there as well. And I wanted to plug our master's program shamelessly.
Yeah.
We are now accepting master students at LaGrange College in Georgia. So we also offer a master's degree online, and it's specifically dedicated to strength and conditioning. So like the focus is human performance.
It's awesome.
It's very heavy on athlete monitoring, sports science, strength and conditioning, program design, periodization. So we dig into that. We have a really cool sport nutrition class that I'm teaching now. So anyone interested in the stuff that I've been talking about and related to strength and conditioning, I would definitely welcome applications. So yeah, man, I think that's it.
Awesome, man. Well, I appreciate you coming on so much.
Thank you, dude. Yeah, and we can hopefully chat again when the review comes out, and we can get into some more like really heavy muscle fist stuff, or talk about like the later half of sort of like a part 2 of what happened from 1980s to now, and then where we're going from there. So I'd be more than happy to do that too.
Yep, I'm totally on board with that. That sounds great.
All right, Ryan. Thank you for what you're doing, man. It's awesome. Keep it up.
Yep, thank you. Thank you so much for checking this out. Make sure to go check out Cody's stuff, check out my stuff if you're interested in coaching and things like that at ryanjsalm.com. Screenshot it, put it on your story, and I'll see you in that next episode.